bacteria divide by binary fission, and they divide very, very quickly.
It creates an anaerobic environment, which means no oxygen.
This is an analytical profile index, and essentially what it allows us to do.
The Division of Consolidated Lab Services, they came into our classrooms to conduct station lessons with our students. So that was pretty much an extension of what we've been covering in our unit for viruses and bacteria.
And a lot of the biochemicals are sugars or amino acids that the bacteria can use as an energy.
Within those stations, they got the chance to look underneath microscopes. They were able to look at the processes and see how the scientists in the field actually use the equipment daily.
You can see this is clear right now. So when we mix it up, we want it to have the same.
From what I observed, the students were really engaged. They enjoyed it. They were asking great questions.
So how do you know what it is?
So there's this.
It was a really valuable experience all around.
Even though we are not working with hazardous materials, we all want to simulate that environment that you see all of us garbed up for appropriately, and that's going to include a water fluid resistant kind of gown or lab coat of some sort, eyewear protection, as well as gloves when you're working at the different stations.
Just the entire presentation, how they arrived and had all of their personal protective equipment, I really think that it really put the students in a scientific mind frame. Like they really in that moment felt like scientists, whereas I can't necessarily with high confidence say that they felt that way before.
I usually hold it flat. I don't try and
My goal and my aim is to expose students to as much as possible and for them to really gravitate towards to whatever their passion is and whatever they want to pursue further interest in.
If it's kind of greenish, we'd say you probably start with negative.
Back in November, the students actually toured the Division of Consolidated Lab Services.
This is the arbovirus section.
We do 2 kinds of pesticide testing.
So to see where that information comes from and how they go about finding out these things and these statistics that we hear about in the media, I think, again, that was another seamless connection.
That's called an autoclave.
We have some other topics that we haven't quite covered yet. One of them is DNA and genetics, so very soon we do plan on meeting with them again and trying to tailor another experience.
To be able to identify what bacteria we're working with.